So continuing from Chapter 12, Chapter 13 is going to talk about DNA synthesis. These are the learning objectives for Chapter 13. Be sure that you can answer these completely. So DNA replication occurs in the S phase of the cell cycle, and that's where in the nucleus one new DNA strand is synthesized from a parental strand. So here's the eukaryotic cell cycle. We just said that DNA replication occurs in the S phase. So all organisms that consist of cells and arise from pre-existing cells. So part of cell division is mitosis and meiosis. If you look at the helix on the right, all new strands are made from the template of the parental strand. So here is the first learning objective, knowing the different terms in DNA replication. Again, DNA replication occurs in the S phase of the cell cycle. The leading strand is going to be continuous, 5' to 3'. The lagging strand is going to be discontinuous, 5' to 3'. Okazaki fragments are the names of DNA strands that are made discontinuously in short segments. DNA polymerase is the enzyme involved in replication. And then DNA ligase is what joins the discontinuous strands together. So DNA replication occurs at the replication fork. That's the Y structure at the very top. If you look at those arrows, you'll see these zigzag portions. Those are your RNA primers. So in order for DNA replication to start, you need an RNA primer to help the DNA polymerase get started. So first round of synthesis occurs. Unwinding of the helix is usually done by your DNA helicase. And then you'll have a leading strand and you'll have a lagging strand. So there's no problems making the leading strand because DNA polymerase can make strands in the 5' to 3' direction. But where we encounter problems is when we go to the lagging strand, which is in the 3' to 5' direction. So since DNA polymerase has a hard time making it in this direction, it's going to have to make them in fragments. So these fragments are called Okazaki fragments. So as you go on with DNA replication, you can remove the primers. Pretty much DNA polymerase says, hey, I got this, you can leave now. And then those gaps on the lagging strand are going to be joined by your DNA ligase. And that's a general overview of DNA replication. These are just more terms to add on with DNA replication. So the first one is your replication bubble. That's somewhere in DNA where the bases pull apart and then the backbone is going to open up like a bubble. So at the end of each bubble, that's where your replication fork starts. So each bubble will have two forks. Direction of synthesis is important. So it occurs in the 3' to 5' direction, but it produces new strands in a 5' to 3' direction. So if you look at a picture of DNA, the leading strand is going to be made from the 5' to 3' direction. So it's producing new strands in a 5' to 3' direction, but it's occurring in a 3' to 5' direction. Here's a picture of your replication bubble. Remember, each bubble has two replication forks, so that means DNA replication can occur at two ends. So take a look at the picture. It might be confusing to look at both strands at once, but know that direction of synthesis occurs in the 3' to 5' direction. So if we're looking at the top strand, we're starting from the left to the right, but when we're producing the copies of the strands, it's happening in the 5' to 3' direction. So here are a few other definitions. So helicase, remember, is the enzyme that's going to unwind the DNA parental strand so that we can make new strands. Topoisomerase is an enzyme that breaks that phosphodiester backbone, but then it rejoins them. And then you have something called a single-strand binding protein, which prevents the strand from coming back together during replication. So the main difference between type 1 and type 2 topoisomerase is type 1 can relax both positive and negative supercoils, and it breaks one strand. Type 2 can relax but not supercoil DNA, and it also needs ATP. So here we have our topoisomerase inhibitors. And so remember, topoisomerases, they help relieve the torsional strain during DNA replication. So without them, the stress on the double helix would be too great and DNA replication would come to a halt. But there are certain cases, let's say if we have a bacterial infection or if we have cancer, we want to stop DNA replication because we don't want the bacteria to grow or we don't want the cancer to spread. So at the very top, we have your antibiotics. So antibiotic drugs target your type 2 topoisomerase. So nalidixic acid and ciprofloxacin are going to be your antibiotics that target type 2 topoisomerases. On the bottom, we have anti-cancer drugs that target type 1 topoisomerase. So irinotecan and topoctecan. So be sure to know which ones are going to target type 2, which ones are going to target type 1, and then know which one's your antibiotics and which one's your anti-cancer. Here are some more anti-cancer drugs to add to the list. Doxorubicin and etoposide. Both are going to target that type 2 topoisomerase. Here we're looking at replication in prokaryotes. So helicases, again, that they're going to break the double helix apart so that replication can take place. Topoisomerase is breaking those phosphodiester bonds and then rejoining them again. So it's sort of cleaning up the mess left by helicase. But the main thing to pay attention to is the single-stranded binding proteins. So those are just going to be present to prevent the strand from coming back together. So the last definition is the action of DNA polymerase. So DNA polymerase is an enzyme that can replicate or so synthesize new DNA or repair DNA. So here we see that the binding of DNA A to a DNA A box is going to initiate replication, and it's also going to form that replication bubble. So after DNA A binds to the DNA A boxes, DNA B proteins, which is helicase, can bind to the origin and begin separating the DNA in both directions. So that creates two replication forks, and DNA C is just a protein that assists with this process, but is released later on. Here we have another overview of DNA replication. So if we follow the boxes, we have single-stranded binding proteins, which keep the strands apart. We have helicase, which breaks those hydrogen bonds in the double helix so that replication can begin. We've got topoisomerase, which follows behind helicase to alleviate the supercoiling. And then we have the primase, which synthesizes that RNA primer that we need to begin DNA replication. So that's going to be removed later, and then DNA polymerase is going to take over for the synthesis of new DNA. So DNA polymerase 1 and 3 do the proofreading. DNA ligase is what's going to join together those Okazaki fragments. And then you have your type 1 and type 2 topoisomerase. So type 2 topoisomerase is also called DNA gyrase. So here is just saying the same thing again. If you look, the removal of the RNA primer leads to that 3' overhang. So chromosome shortening eventually leads to cell death. So the way to solve this problem is to add a telomerase. It then acts as a template for the rest of the DNA strand. So the original length of the chromosomal DNA has been restored, and now the gap where the primer for DNA replication has been removed. So here's another learning objective, is knowing the similarities and differences between eukaryotes and prokaryotes. So really know the differences between them. He'll test you on how well you know them. So here's another important slide. It's a table. Notice the protein and then their function. So we talked about several of these already. Single-stranded binding proteins, remember, are going to bind to a single strand of DNA to prevent them from coming back together. DNA B protein we talked about was helicase, which is going to unwind your DNA. Your primase, also known as DNA G, is going to prime for synthesis. Then you have DNA polymerase 3, which is going to synthesize that new strand. DNA polymerase 1 fills in gaps, and DNA ligase is going to put together those Okazaki fragments. And then DNA gyrase, also known as topoisomerase 2, is going to relieve the torsional strength. Here again is just another table, very similar to the one we just looked at. So be sure to know those different proteins and their functions. So here's another drug called norfloxacin. It's a member of the fluoroquinolone family, and it's used to treat urinary tract infections. So norfloxacin is going to inhibit bacterial DNA gyrase. So remember, that's type 2 topoisomerase. So this again is just showing you that polynucleotide structure of DNA. So remember, DNA is made of 3 subunits: your deoxyribose sugar, your base, and your phosphate. And then remember, nucleotides are joined together by a phosphodiester bond. So the whole backbone of DNA is a phosphate backbone. So this is just showing you what the 5' end of the first nucleotide looks like. This is just showing you the action of DNA polymerase. Remember, your DNA template strand is in the 3' to 5' direction, but new strands are being synthesized in the 5' to 3' direction. So parental strand is 3' to 5'. Your growing new chain is going to be 5' to 3'. So here's just another recap of DNA replication. Hopefully you understand most of this by now. If you need a visual, I put another link to a video that will explain most of this. So benzo-A-pyrene is something that is found in cigarettes. It's only carcinogenic when it's oxidized within the cells. So when it is oxidized in the cells, it's going to bind to your guanine, and it's going to distort its structure. So guanine normally base pairs with cytosine, but now you have this distorted guanine structure, so it's going to produce distortions in the helix. So here is another example of something that could distort the helix. With ultraviolet light, you can cause 2 adjacent pyrimidines, so thymine in this case, to form a covalent dimer. So thymine usually base pairs with adenine, but if it forms this dimer, then it's going to cause problems in the helix. Here again is just showing you the same thing from the previous slide, formation of that pyrimidine dimer, which is caused by UV light. So here again is showing you the dimer that is being formed by UV light, and it's showing that it makes a kink in your double helix. So dimers can be repaired by photoreactivation or nucleotide excision repair, but unrepaired dimers are going to be mutagenic. So these are the primary causes of skin cancer in humans. I'm going to switch over and talk about repair mechanisms to fix problems with the DNA helix. So at the top we have a normal DNA. The second picture shows the damaged DNA. So what can we do to fix it? We can use endonucleases, which are going to cleave the phosphodiester bond within the polynucleotide chain. So that third picture, you see that the damaged region was removed. We can also use exonucleases, which are enzymes that cleave nucleotides one at a time from the end of the chain. So after the damaged region is removed, repair DNA polymerase is going to synthesize a new strand, and then DNA ligase is just going to join it together. Some other repair mechanisms are base excision repair and nucleotide excision repair, which we'll see in the next slide. So on the left we have base excision repair. So in base excision repair, a glycosylase is going to cleave that glycosidic bond between the damaged base. So it's removing the damaged base, and then an incision endonuclease is going to cleave the strand. And once all of that's done, all the different enzymes like your DNA polymerase and DNA ligase are going to repair it and restore the region to normal. So on the right side, we have nucleotide excision repair, and it's very similar, except in nucleotide excision repair, the entire nucleotide is removed at once instead of just a single base. So both of these mechanisms are using excision endonucleases to remove the damaged or distorted region, and then the gap is filled by DNA polymerase, which makes the synthesis of the new strand, and then DNA ligase is going to glue them back together. In addition to nucleotide and base excision repair, we also have mismatch repair, which during replication, if it encounters any bases that are mismatched, then the mismatch repair enzyme complex is going to recognize it. There's also transcription coupled repair. So during transcription, if RNA polymerase stalls because it encounters a damaged region of DNA template, then the transcription coupled repair complex will take place. So in bacteria, we have parental strands that are methylated, and we have the newly synthesized strands, which is unmethylated. So your newly synthesized strand could contain a mismatched base, and this is how the different proteins would recognize it. They would recognize the parental strand because it's methylated, and then they would just replace the unmethylated strand with a new segment of DNA. So here we're going to talk about genetic rearrangement, so recombination and translocation. Key steps in recombination. Segments of DNA can be exchanged, and this happens a lot in DNA, but this leads to genetic alterations, and it can either be beneficial or devastating. So here we have two homologous DNA molecules. What happens to number 2 is it's being nicked, and then it invades the other homologous molecules. So they're going to go through nicks, and they're going to exchange their segments and form something called a holiday structure. So once the holiday structure is formed, it'll twist and isomerize, it will be cut, and then it will be resealed, and then you have your new DNA molecules. So sometimes there are breaks in chromosomes, and that's usually caused by X-rays or chemical carcinogens, but this results in chromosomal translocation. So the free end of one chromosome is going to have a break point, and it's going to reseal with the free end of a different broken chromosome. So in this case, the normal chromosome 8 is going to find and reseal with chromosome 14. And so sometimes these exchanges are going to have detrimental effects, and they're frequently observed in cancer cells. So here are the steps involved in transposons. So transposons are known as jumping genes. They're segments of DNA that can move from their original position to a new location. And transposons do this by using this enzyme called a transposase, which will cleave that DNA strand. So if you look at the picture, you have a DNA strand, and a staggered break is produced by that enzyme transposase. So this allows for the transposon to integrate itself into the DNA, and once that happens, repair of the gaps is going to occur, and you'll also notice those regions in blue are just direct repeats that have the same base sequence in the 5' to 3' direction. So reverse transcriptase is just an enzyme that uses single-stranded RNA as a template to make a DNA copy. So if you look at the picture, we have a single-stranded RNA template, and that's going to make something called cDNA. So cDNA stands for complementary DNA because it is complementary to that RNA strand. So once we have our cDNA, the RNA template is going to be degraded, and then we can make another strand using that cDNA template. Here's the life cycle of a retrovirus. It also contains a reverse transcriptase. So once the virus enters the cell, it's going to use reverse transcription to make that same double-stranded cDNA that we learned about in the previous slide. So once we have our double-stranded cDNA, it's going to be integrated into the human genome, and then it's going to start replicating. So here are some antiretroviral agents and the mechanism. So be sure to know each of these. For example, raltegravir, it targets integrase, and integrase is an HIV enzyme. So if it's targeting this HIV enzyme, it's going to inhibit integration of your viral genetic material into the genome. So here's the life cycle of HIV. Be sure to know those drugs and what they block. These are the same drugs that you saw in the previous slide. So, for example, he can ask a question, what blocks the fusion and uncoding of a virus? So that would be blocked by your fusion inhibitors, which is enfervatide. So here are your structures of the drugs. I'm not sure if he's going to test you guys on it. He didn't for us, but it seems that he is changing and integrating more structures this year. So this is just showing synthesis of zidovudine, and it was originally intended to treat cancer, but it failed to show efficacy because it had a highly toxic profile. So here is our infamous disease table. Be sure to read through those disorders and know their condition details.